we talked about the transfer. Now we're gonna it's gonna be a desperate attempt in a very short period of time to figure out how the expression is regulated. Because remember when we talked about gene expression, we mentioned that not all the genes are expressed at a given time, right? So there should be something that controls that expression. So there are two big mechanisms. I'm not saying that those are two only mechanisms, but two mechanisms that I want you to be aware about. The mechanisms are operons that control gene expressions and riboswitches. Now the operon is the name of the fragment of a genome that's usually responsible for some kind of a trait, some kind of a function that contain, and this fragment of the genome, the operon, usually contains several genes. Does that make sense? Now look, gene encodes for a protein. This protein can be an enzyme, right? Does that make sense? It can be a regulatory molecule. It can be something else. But the point is, often for a gene, for a trait to be present, to be expressed, for a trait to be observed, for a specific phenotype, you have to express more than one gene. Does that make sense? If you, well, and I believe you have taken the biology in one or another form, whether in high school or here, you have heard about Mendelian genetics, right? His experiments with peas and stuff like that. Now, in humans, for example, traits, phenotypical traits that we can observe are often not controlled by one gene, but multitude of genes. Does that make sense? For instance, human height, how tall we are, that is determined by several genes. Does that make sense? That's why it's not like we have only really tiny or really tall people. We have all different heights. Okay? Now, you can say that it is some sort kind of operon, although operons are used only in relation to the bacteria and archaea, not in eukaryotes. So the example of the operon that we're going to discuss later are operons that control utilization of lactose as the energy and nutritional source by microbes, and operon that controls the synthesis of amino acid tryptophan. So if you look at the structure of the operon here, you will see that actual genes of the operon are controlled by the number of other sequences. The promoter sequence. What is the function of promoter? What interacts with the promoter? Does anyone remember? Which enzyme binds to the promoter to start gene expression? RNA polymerase. Very good. RNA polymerase binds to the promoter, right? Now, operator sequence right here, okay, controls whether RNA polymerase can proceed or not. Does that make sense? Not exactly. Promoter marks the beginning of transcription. Does that make sense? Does that statement make sense? Promoter marks the beginning of transcription. If you're a runner, you know, there's a start line. Okay, so that marks where you start the race, right? No. I'm talking about so if you talk about codons, you have start and stop codon, that determine the beginning and the end of translation. Wait a minute. Codons mark the beginning and the end of a protein. Does that make sense? Does it? Okay. Promoter marks where the transcription begins. That's where RNA polymerase binds to. Okay? Is that clear? Now, whether RNA polymerase will be able to actually transcribe depends on the operator here. Imagine that you're a runner and you are at the starting line. You're about to run, and then suddenly somebody puts the fence in front of you. Will you be able to run through the entire distance? Well, unless you jump that fence, but imagine that you can't. So that's it. You have all the capacities to run, but you can't because you have a fence in front of you. So the task of operator sequence is to control, well, it's pretty much what operator does through several mechanisms. It controls whether RNA polymerase can proceed with the transcription or cannot. That's the place where you build defense. Does that make sense? Now, if you would look at the following, the structural genes. Remember we mentioned about the polycistronic RNA versus monocistronic. Do you remember what the difference is? What's the polycistronic mRNA? Poly means many, and it contains many genes. Remember? So this is poly. Let me switch to another color. It's kind of stupid. Polycistronic mRNA, because it contains A, B, and C and D genes, right? A, B, C, and D. It's all one long transcript. That makes sense? You see that? And then during translation here, they produce separate proteins. Because each protein now has a start and stop codon. Sequence of each protein in mRNA starts with AUG and ends with a stop codon. Does that make sense? You sure? Say again? Oh, there is a termination sequence at the end of the operon. It's just some kind of a signaling sequence that tells RNA, well, pretty much terminal repeats. So RNA polymerase just falls off. Okay? That marks the end of the operon. Like the transcription itself, it's the same. The entire process is the same. We talk not about the transcription per se, but how we regulate whether to give it a go or not. Does that make sense? Okay, so one thing is the operator, okay? Sometimes you have some regulatory genes in a different location that produce certain proteins that will regulate the function of the operon, okay? Now, another system is quite interesting. It is called riboswitch. It looks horrifyingly complicated, but it's actually, on the conceptual level, is a very simple concept. So remember I mentioned that there are certain structures that can exist in the single-stranded RNA molecule. I especially focused on the hairpin loop structure. Remember? This is the hairpin loop. Okay? So these structures often regulate whether transcription or translation can or cannot occur. So this is the example when special structure called anti-terminator allows RNA polymerase to transcribe a gene. Okay? Upstream of anti-terminator loop is the RNA structure that is called riboswitch. If a small molecule regulator, usually it's a small organic molecule, it can be a molecule responsible for quorum sensing, molecule that is produced by other microbes when they grow to a certain concentration. We talked about it a little. When it binds to the riboswitch, it changes the riboswitch structure and changes the structure of the anti-terminator stem loop, which is now becoming terminator stem loop. Because of the changed structure, RNA polymerase cannot bind to the promoter anymore, and transcription is off. Does that make sense? Think about riboswitch. It's the light switch. That's it. That's all it does. Not this fancy one, but the old-fashioned, when they go up and down. Because what you essentially do when you turn on the light, you move some thingy inside of the light switch, right? And that's it. You mechanically move it. And it's you that have to come and switch it. Otherwise, the light's going to be on. So this is you. This is you that comes over and flicks the switch. Yeah, riboswitch in one position. So in the on situation, this stem loop allows the transcription. When molecule binds to riboswitch, some kind of organic molecule binds to the riboswitch, anti-terminator becomes terminator, and transcription stops. Does that make sense? It's simple on-off regulation. It even cannot bind there. It cannot even start. With anti-terminator, it just goes, yeah. See, okay. This shows you the situation when molecule inhibits the transcription. Does that make sense? So when molecule binds, transcription stops. Can it be the opposite way? Yes. Does that make sense? So riboswitch can turn off, or it can turn on. Molecules can either turn on the transcription or turn off the transcription. Does that make sense? Same goes for the translation. You can have riboswitch for the translation. Okay? So when molecule binds to the riboswitch, RNA structure changes so that binding site for the ribosome becomes structurally blocked. Ribosome cannot bind to mRNA, and there is no translation. That make sense? The difference between riboswitch and the operon structure. Operons exist in the DNA. Riboswitches are RNA elements. Does that make sense? So riboswitches are less profound. more fine-tuned. Okay, think about, again, for riboswitches, think about them as on-off regulators of transcription and translation. Okay, that alter the structure of either the transcription terminator and T-terminator loop or ribosome binding site. Operons have more fundamental effect, and that's what I want to talk about now. So operons sorry, can be inducible or repressible. So when we say inducible operon, what it means that there is some sort of a signal, some sort of a chemical that can initiate transcription of genes in the operon. It sounds really not English, but here how the inducible operon works. And we're going to use as an example the lactose operon, probably one of the most studied bacterial genetic elements in science. Lactose operon consists of several genes that produce enzymes necessary for utilization of lactose, either as the nutritional or as the energy source. Normally, transcription of these genes is repressed. Protein molecule called repressor sits on the fragment of the operon that is called operator, the one that we just saw. So even though RNA polymerase can bind to the promoter, it cannot move forward. It cannot transcribe those genes and initiate the gene expression because repressor is in the way. Does that make sense? That's the runner with the fence in front of him. Does that make sense? When microbe encounters lactose in the environment, lactose molecule binds to the repressor, and complex of repressor with the lactose molecule dissociates from the DNA. There is no blocking of transcription, and RNA polymerase can now proceed and transcribe the genes. Does that make sense? Pretty much lactose removes that fence. For lactose operon, only lactose. For other operons, it can be something else. For instance, there is a ribinose operon that works pretty much the same way. When there is a sugar called ribinose in the media, it will bind the repressor. Repressor will fall off the DNA, and transcription of the genes responsible for ribinose metabolism will start. Does that make sense? You sure? Okay. Yes. Mm-hmm. Wait. You're running too far, too fast and too far. I think I know what you're talking about, and you will tell me if I will answer your question now. So, now let's write down very basic chemical equation. It's not even chemical. What happens to lactose? So I have lactose, which is converted into, I don't know, energy. Okay? Right? It is metabolized, and that metabolism is catalyzed by enzymes, right? What produces those enzymes? which part of the bacterial DNA encodes for these enzymes? These ones. Come on. The answer is right in front of you. Say it. Inducible lactose operon, right? So these enzymes are encoded by these genes. Does that make sense? So in this case, the substrate of enzymatic reaction regulates the operon. When those enzymes are produced, what's going to happen to the lactose levels? They're going to go down, right? Because you saying? Yeah, you start with a lot of lactose, which initiates the expression of genes, the lactose operon. Enzymes will metabolize the lactose, and lactose levels will go down. When lactose levels will go down, what's going to happen to that complex? Yeah, it's going to stop existing because there's no more lactose. So repressor will bind back and will stop the production of enzymes. Does that make sense? It's the classic negative feedback regulation. So inducible operons are stimulated by the substrates of enzymatic reactions. Now, let's take a look at the repressible operons. The tryptophan operon. Tryptophan is an amino acid that is required for the normal growth of microorganisms, blah, blah, blah. Okay? Normally, this operon works. Okay, so RNA polymerase transcribes the genes that are necessary for tryptophan synthesis. These genes produce tryptophan, more and more and more tryptophan. Eventually, when levels of tryptophan in the cell reach certain level, tryptophan binds to the repressor. And that complex of repressor and tryptophan will inhibit the transcription from the tryptophan operon. Does that make sense? Let's write the reaction. You have chemicals, tryptophan in the end, and enzymes. What produces the enzymes? What encodes for these enzymes? Tryptophan, or it's not inducible. Tryptophan, we call it tryptophan operon. But look, when tryptophan accumulates, it represses the operon. Does that make sense? It accumulates, it represses the operon. Then bacteria uses all the tryptophan. Tryptophan levels fall down. What's going to happen to that complex? It's pretty much going to disappear. Repressor will fall off the operator, and the expression of the genes will start over. Does that make sense? So, inducible operon is induced by the substrate of enzymatic reaction. Repressible operon is repressed by the product. Think about from economical standpoint. When company produces some kind of a product, if it's too much, they will stop making it, right? Because they just can't sell it. Nobody's going to buy it, right? And then when everybody uses that product, they will start to produce it again. Does that make sense? So that's the entire, but you have to understand how these operons work. If for riboswitches, all I want you to know is, it's on-off regulation of transcription and translation, and that regulator is located in mRNA, then for operons, I will ask you, what inhibits, what represses in repressible, what induces in the inducible. I will give you a description of how some generic operon works and you will have to tell me whether it's inducible or repressible. Okay? I will give you a description and you have to tell me this is substrate and I don't know, it induces, or this is the product and it suppresses. Does that make sense? So please know this stuff. Okay?